Okay, let's dive right into this explainer. Today, we're going to discover how a microscopic cellular copying glitch scales all the way up to become the absolute ultimate forensic tool for human identification. It's essentially a biological detective story, and it's happening inside your cells right now.
We've got a fascinating journey ahead, going straight from the invisible molecular level to massive FBI databases. We'll quickly cover what satellite DNA actually is, break down that microscopic cellular glitch, see how harmless mutations turn into polymorphisms, sort some DNA with electricity, and wrap up with the blueprint of fingerprinting.
So, to kick off our micro to macro journey, we first need to understand the fundamental building blocks. Section 1. What is satellite DNA? The truth behind the name. Now, you hear a term like satellite DNA and you immediately think of space, right? NASA, satellites orbiting a planet, that whole thing. Well, it actually has absolutely nothing to do with space. It's entirely based on how the DNA behaves in a laboratory centrifuge. You see, when scientists first started isolating DNA, they needed a way to separate these massive blocks of repeating genetic code from the rest of the genome. Imagine spinning genomic DNA in a high-speed centrifuge filled with a dense salt solution. The DNA physically separates based purely on its density. Since most human DNA is a pretty balanced mix of base pairs, it all settles together into this one massive, heavy main band right at the bottom of the tube. But there are these repetitive sequences that are highly skewed. They are so rich in physically lighter adenine-thymine pairs that they separate completely from the heavier bulk of our genome and they end up floating higher up in the tube.
And this is exactly where the name comes from. When researchers mapped out the density graph of that centrifuge tube, those distinct subsidiary bands floating higher up literally looked like tiny celestial satellites orbiting a massive planet. So it's just a really beautiful visual metaphor for a purely chemical reaction.
Okay, let's move on to the inciting incident of our whole story. Section 2, the microscopic cellular glitch, and how our cells miscount.
So, how do we get these varying repeats in the first place? Well, it all comes down to a really quirky biological process known as polymerase slippage. Think of the DNA enzyme like a tired scribe trying to copy a book by hand. When it's reading a normal sentence, it's totally fine. But when it hits a monotonous repeating sequence like GATA, 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 it gets confused and literally loses its place.
During that split second of confusion, the newly synthesized strand slips backward, unpairs from the template, and forms this tiny temporary loop. But the polymerase enzyme is completely unaware that it just hiccuped, so it keeps right on copying to the end. The final result? When the DNA repairs itself, that little loop is locked in. The new strand permanently contains an extra repeat unit that the original template didn't even have. Or conversely, if the template slips, a repeat gets deleted.
The absolutely crucial point here is that this cellular glitch acts as a physical engine. Every single time our cells divide, there's a chance for this engine to run, constantly shuffling the length of these repeating DNA blocks. It is essentially a biological typo factory that makes these specific regions highly, highly unstable.
Now we have to ask the big question. Section 3. Harmless mutations become polymorphisms. How does a random glitch in a single cell survive long enough to become a global forensic tool?
Before we get too far ahead of ourselves, let's quickly define our terms. A polymorphism simply means a specific genetic variation has survived and multiplied enough to exist in more than 1% of a population. And in this context, we aren't talking about a change in the genetic letters themselves, like turning a G into a C. We are talking about a physical change in the length of the sequence due to a totally different number of those repeats.
Now, you might be wondering, wouldn't these constant mutations make us sick? Well, if a slippage glitch happens inside essential, coding DNA, it usually alters a vital protein. And natural selection quickly eliminates those changes because they threaten our survival. But these microsatellites, they're located in non-coding zones, which we historically call junk DNA. Because changes in these zones have zero effect on our health, these constant copying glitches are completely harmless. They just accumulate wildly over thousands of years, totally unchecked.
What starts as an isolated copying error, let's say in a single sperm or egg cell, eventually cascades down through the generations. A child inherits that unique sequence length, and as their descendants multiply over centuries, that new length spreads. Once it crosses that 1% threshold, it graduates to a stable polymorphism. This results in an enormous pool of different lengths across the human race.
And this high variability is exactly why this matters so much. It provides the unique genetic variants across individuals that make identification possible in the first place. Think about it.
If human DNA didn't tolerate these harmless mutations, we would all look exactly the same at the molecular level. Forensic science as we know it simply would not exist.
So we have all these different DNA lengths safely hidden inside our cells, but how do forensic scientists actually get a look at them? Section 4, sorting DNA with electricity, visualizing the invisible code.
We know the repeats are there, and we know everyone has different lengths. But how on earth do we turn these invisible microscopic glitches into a readable visual barcode that you can actually present to a jury in a courtroom? Well, scientists use a truly fascinating laboratory technique called gel electrophoresis.
Here is the sheer genius of this process. Scientists don't bother testing your whole genome. First, they use chemical clips called primers to isolate just the target microsatellite. Then they use PCR, which is basically a chemical Xerox machine, to amplify that tiny fragment millions of times over. Now that they have a workable sample, they carefully load those amplified fragments into tiny pockets at the end of a porous slab of gel. And then they throw the switch.
The moment they throw that electric switch, basic physics takes over. Because the phosphate backbone of DNA inherently carries a strong negative charge, the current repels it. The DNA fragments desperately try to migrate away from the negative electrode and move toward the positive one. To do this, they have to push their way through the gel matrix, which acts exactly like a molecular sponge filled with microscopic pores.
It's literally a microscopic obstacle course. The short fragments, the ones with fewer microsatellite repeats, slip easily through those tiny pores, flying all the way down to the bottom of the gel. But the long, heavy fragments with tons of repeats, they get all tangled up in the matrix. They move very slowly and get stuck near the top. By just applying electricity, scientists physically sort your DNA based entirely on how many glitches your ancestors accumulated.
Once they turn off the electric current, they apply a fluorescent dye. When you shine UV light on it, those sorted lengths glow as distinct bands. And keep in mind, you inherit one set of chromosomes from each parent, so for any single targeted location, you're usually going to show exactly 2 glowing bands on that gel. It creates an individualized, barcode-like pattern inherited directly from your biological mother and father.
Let's move to our grand finale and see how this entire sequence builds the absolute foundation for modern law enforcement. Section 5, the blueprint of fingerprinting, the ultimate chain reaction.
When you step back, this entire explainer really boils down to one unbroken chain reaction. It starts safely in the non-coding regions of our genome, which allows for a high rate of harmless mutation. That mutation creates widespread DNA polymorphism across the human race. That polymorphism means we all have different fragment lengths. And finally, those varying lengths give us our totally unique gel band patterns when we expose them to electricity.
It's amazing how perfectly the biological components map directly to the technical tools. The polymerase slippage is the physical engine creating the variation. The microsatellites, specifically short tandem repeats or STRs, are the exact targets law enforcement goes after because of their extreme variability. The PCR amplification acts as the Xerox machine for a tiny speck of crime scene evidence. The gel electrophoresis sorts it all out by size, and finally, all of this data feeds directly into CODIS, the FBI software network that tracks these exact length variations to instantly match crime scene evidence to convicted offenders.
It's absolutely wild to think about. We've gone all the way from a tiny enzyme losing its place in a string of letters to a massive national database bringing criminals to justice. So the next time you hear about a forensic DNA match on the news, will you remember the microscopic cellular glitch that made it all possible? Because at the end of the day, the single most powerful tool we have for solving crimes relies entirely on ourselves occasionally forgetting how to count. Thanks for joining me for this explainer, and keep questioning the incredible science hiding right inside you.